This has been a year since we interviewed you at the ARVO 2015, and you presented a new paper here today.
Yes.
Please tell us about that paper.
Okay. Well, my work last year analyzed the amyloid precursor protein, and what we found in that work was that it has polarized expression in the retinal pigmented epithelial cells, which are considered the barrier cells of the eye. This was very significant because we saw this polarized secretion and expression of APP under hypoxic conditions. And what this did was in the apical direction, or going into the eye, APP secretion vastly diminished, and going out of the eye in the basolateral direction, we didn't see this diminishment. So we followed up on these results by looking at various different byproducts that APP also produces as it is cleaved. And the ones we've looked at are SAPP alpha and SAPP beta, two byproducts from the different cleavage pathways of APP. The alpha byproduct is believed to have potential beneficial effects on cell growth and motility and maybe health as well. And the SAPP beta byproduct is considered to be not as helpful because it's a byproduct from the amyloid beta production pathway of APP. And in studying these byproducts, what I did was, in my previous work, I had knocked down the retromer protein, which is a transport vesicle for APP. And when I did that, APP expression and secretion also diminished. And in knocking this down, looking at these specific byproducts, we saw that the SAPP alpha, or potentially beneficial byproduct, did not decrease. But the potentially harmful byproduct, SAPP beta, did decrease. And so this has implications showing that perhaps retromer and APP do indeed continue to co-localize during hypoxic events. And maybe by knocking down retromer, you're not allowing these harmful cleavage proteins and enzymes to interact within retromer, and you're not having these harmful byproducts. To build on this research, we've also been trying to understand how in hypoxia the APP pathway is specifically affected. So we've undergone preliminary mechanistic studies. And what we have done here is done lysosomal and proteasomal inhibition with chemicals. By doing these, it is known that APP is degraded in the lysosomal pathway. When I applied lysosomal inhibition to my cells, we didn't see APP degradation internally. This was not surprising because that's where it's believed to be degraded. What was surprising was when we applied proteasomal inhibition, we saw a complete loss of APP in these preliminary studies. This was very significant to us because this was very unexpected. We've seen this in not only our non-polarized cells, but also our polarized cell model, which has a more in vivo-like nature in the results we can achieve. And while we're still studying these results and trying to build upon them to understand this pathway more, we believe what may be happening under hypoxia is that the RPE could be undergoing an unfolded protein response, or perhaps endoplasmic reticulum stress, thus not allowing APP to be properly degraded and shunting it into other degradation pathways, or degrading it earlier than it should be. These results have been very intriguing, and we continue to build upon them in further studies.
What is your next study to follow up on this?
Our next study is we want to know is maybe this isn't an unfolded protein response or ER stress, so we've also been investigating the autophagocytic pathway. And so I plan to apply autophagocytic inhibitors to not allow APP to potentially be degraded in that pathway when hypoxia is induced to the cells.
What is your follow-up to that? I mean, if you take it to its logical conclusion.
So the follow-up to this is, well, since we know that hypoxia is affecting APP production, we ultimately want to know how is this obviously changing the expression and secretion that we have seen, because we believe that in drusen formation, which is going in the basolateral direction of the RPE, we know that as APP is secreted and it is cleaved into these amyloid beta byproducts, that this could be potentially contributing to that drusen formation in AMD. So the follow-up to these studies would be to also study the accumulation and secretion of amyloid beta in the lysates and in the secretion pathways to see if hypoxia is affecting this as well.
Well, amyloid beta is found in a number of diseases.
Yes.
Not just in the eye. Are there implications across other diseases where amyloid beta is expressed or found?
The biggest implication would be within the brain, since obviously amyloid beta is involved in the Alzheimer's disease pathway, and hypoxic events can also occur within the brain as well. So we believe this research can be translational and not only to be involved just in the eye, but also in Alzheimer's and maybe potentially other diseases throughout the body.
When we look at potential therapeutic measures, events or activities that could interrupt this cycle.
Yes.
Have you thought about some of those?
The main therapeutic measure that we have considered would be to somehow maybe manipulate, again, these degradation pathways that I've talked about that we've disrupted through chemical inhibition, but maybe potentially as well the retromer pathway. Because since we have shown that when you knock down the retromer expression of that protein, it then changes the APP effects of cleavage. Potentially, if you maybe strengthened the retromer protein or degraded it, you could have implications on the APP pathway.
So this really could have quite dramatic translational implications as it turns out.
Yes.
Excellent. Do you tell us about any other work that you're doing that is of interest that we should be aware of?
The other work we're focusing on in the lab is APP is believed to be involved in iron export in the cell, because our lab also studies the iron metabolism of the eye, because that is also involved in hypoxic events and ocular diseases. What has been shown by other labs, and our lab is currently investigating this, is that APP and ferroportin, which is an iron exporter, can co-localize at the cell membrane. When this co-localization occurs, it is believed that APP then strengthens ferroportin and can hold it in place and allow more iron to be exported in the cell. So when a hypoxic event or other type of ocular damage occurs, this excess iron can then leave the cell and not have as much degenerative effects.
So have you looked at it in situations with different concentrations of iron?
We haven't looked at different concentrations of iron yet. We've mostly been studying how APP and ferroportin can co-localize. Once we have confirmed that we have co-localization within our cells, we do plan to do dose-dependent studies and things of that nature to see how the effect.
But there's a clinical association that you probably know in patients that have hemochromatosis or iron overload, and a variety of pathological retinal events. And I know there are some clinicians working in this space. So it'll be exciting to see what that particular line of research that you're doing brings forth.
Yes, we hope it does evolve into a translational study for sure.
Excellent. Philip, thank you so much for coming by and giving us all the good news, and we look forward in anticipation to your next publication.
Thank you very much.
Thank you.